Welcome to the Public Affairs Show of your favorite local iHeartRadio station in Seattle. I'm your host, Lee Callahan. Got two great guests on the phone with me today from two different local organizations working together, partnering to make life better for all of us. From Ozette Technologies, I have Cherie Green. She is the Vice President of Translational Science at Ozette. And from Bloodworks Bio, part of Bloodworks Northwest, I've got Executive Vice President Aaron Posey. I'll start with Aaron. We're all familiar with Bloodworks Northwest. I've had your CEO on many times to talk about blood donation and how critical that is. But Bloodworks Bio is a portion of what Bloodworks Northwest does. Will you tell us about that?
Definitely. Yes, I'm Aaron Posey, the Executive Vice President of Bloodworks Bio. We are a department within Bloodworks. And we provide starting material to cellular and gene therapy organizations to fuel their pipeline of drug development. And so what does that actually mean? So we provide different types of blood products, very similar to the main operations of Bloodworks Northwest, where we supply healthy blood for transfusion. We also start with blood, but we take it to a different level, if you will. We can remove from blood, either it be whole blood or blood derived from a process known as apheresis. Mm-hmm. We can remove specialized cells per the specifications of our cell and gene therapy customers. We can then enrich those cells or make them a homogeneous population to provide to the cell and gene therapy companies that they use in their platforms for generating cell and gene therapy drugs or how would I put that?
Pharmaceuticals, medication?
Yeah, you got it. Yeah. Yes. Cell and gene therapies across, many different ailments, including, cancer, autoimmune disease, etc.
Wow. Okay, this is good information. Many questions are a-brewing already. I have got Cherie Green. She is from Ozette Technologies. Would you tell me about what Ozette does? You're right here in Seattle.
Absolutely. Ozette is a startup company that was founded in the heart of the Pacific Northwest in Seattle, and our technology really focuses on innovative approaches to profile single cells, so the immune system, what kind of cells are in our body. And our technology originated from the Fred Hutch Cancer Center, with leaders in the field of computational science. And our founders include Greg Finnack, Rafael Guittardo, and Evan Green. And so what we've done is we've spun out a company to really focus on using state-of-the-art technologies such as flow cytometry and RNA sequencing and machine learning algorithms to analyze the data with the mission of understanding the immune system, both in health and disease. And really our goal is to, deeply profile and understand the immune system to accelerate new developments of therapies for a myriad of diseases.
How interesting. Would you say a lot of what you're looking at as far as diseases go are autoimmune, or is it just everything?
Well, I'd say, immune cells are the foundation of our body, and they can be in a health state or in a disease state. And so, this is true for autoimmune, where our immune cells aren't functioning properly. And also in the case of oncology, where you have, cancer cells that overtake our normal or our healthy immune system. And so we really focus on all the cells in the body across many different diseases, including infectious disease, oncology, and autoimmune diseases.
Fascinating. I have got Aaron that's Cherie Green from Ozette. Aaron Posey is on the line also from Bloodworks Bio. Aaron, will you tell me why you two need each other in this partnership?
Absolutely. I think it's actually multifaceted. When we did the ideation phase of these projects, we came up with a number of different ways to think about AI or artificial intelligence to help us solve things that we cannot solve in a laboratory setting. So it's kind of peeling back the onion to really look at other methods that provide the answers to problems that we can't generally solve just by doing laboratory or bench work in a laboratory setting.
Okay. Next, you mentioned apheresis.
The apheresis model can be used for either therapeutic reasons.
Yep.
So when people need a plasma exchange, they will undergo an apheresis treatment for that.
Okay.
You can also derive plasma from an apheresis model for recovered plasma, for fractionation. They could also be used for a growth factor within cellular culture. But what we're really focusing on is really the cellular components that you get out of an apheresis process in abundance, where you can't get them from a typical blood draw.
Okay. This is where the public comes in, because you still need donors for this work that you are doing with Ozette Labs. Am I right?
That is correct.
Yes.
The donors are the basis for what we do. Without donors or without specific donor recruitment tactics, we wouldn't have a business. So every client that comes to us that is in need of a leukapheresis, which is derived from the process of apheresis, we have to go through a very specialized recruitment of not only identifying the donor, but screening the donor for infectious disease markers or, whatever specification the client has asked us to screen for. And then we have to do a vein assessment to make sure that they're suitable for the apheresis procedure. So it's quite a commitment from a donor to take time out of their day to come in and go through a 3 to 4 to maybe sometimes 5-hour procedure so that we can provide the correct material to the cell and gene therapy companies that are generating cell and gene therapies.
Oh, wow. That's a lot longer than it took me.
Yeah, there's quite a few different methods for apheresis, one of which we're evaluating with Ozette. So the most basic is a leukapheresis collection. That's typically around a 3-hour process.
Okay.
And basically, as you had mentioned, your blood is taken from your body. It is put through a machine and a centrifugation process where we remove the components that we're looking for, in this case, PBMCs, or peripheral blood mononuclear sites. And then everything else is returned back to your body, including the plasma, the serum, with a little bit of additive plasma or serum into the leukapheresis pack. But you retain all of the other types of cells. Your red blood cells go back in, most of your plasma, most of your serum, and then your body will regenerate those cell types over time. And so it's a very regulated process.
Cherie Green is on the phone with me also. That was Aaron Posey from Bloodworks Bio, an arm of Bloodworks Northwest. Cherie Green is from Ozette. Cherie, if you could encapsulate, if you were talking to a donor, what would you say that might just push them into coming in for 3 or 4 hours? What would you say? If you do this, then this is going to happen.
Yeah, I mean, I think it's pretty critical for donors to come in and volunteer their time and their precious biological material. It's used in a lot of different ways. One additional, so we've been talking about this apheresis. There is also, a process in which you can enrich specific cells that are important in let's say, transplantation. So cancer patients go through stem cell transplantation. And so there are certain types of cells within that apheresis that can be mobilized through the administration of growth factors. And one thing that Aaron and I are doing together is trying to predict what types of native cells or a composition of cells in the body could predict a better mobilized donor. So stem cells are kind of the beginning path of our hematopoiesis and important in regenerating cells in the body. And so you can infuse stem cells back into a patient for treatment.
Cool.
And what our technology does is it actually profiles all the cells that come out of a product, whether it's whole blood, apheresis, mobilized blood. What's a really powerful component of our collaboration is that, Bloodworks provides these very important blood products for, for patients and for drug development companies. These products are also like the starting material for cell therapy products. So I'm sure you've heard in the news about, CAR Ts. These are a specific kind of cell therapy. And you can use T cells from the patient or from donors, and that's called an allo CAR T. And the material that is generated at Bloodworks Bio is actually the beginning material. And without understanding what the immune profile is of the cells that are going into that starting material, kind of an unknown. So that's where we're excited about collaborating. I was just going to break it down. So, when we talk about cellular therapy can be anything from, taking, stem cells and doing a transplant.
Okay.
That's like, that's been around for many, many decades. But in the last, 10 years, CAR T products, which are chimeric antigen receptor T cells, so they're taking cells from either a healthy donor or the cancer patient, and they're modifying them in some way to be killing machines.
Oh,
so they are potent therapy to target cancer cells in the body and have had pretty sizable responses in certain populations of cancer. And so these are all originating from blood products, either from the patient or from healthy donors.
From Bloodworks Bio, I've also got Aaron Posey. What were you going to add to that, Aaron?
No, I mean, Cherie is exactly right. And, we support all of those clinical trials with a lot of different organizations that are developing the products from the starting material. And, I kind of want to focus on one of the outcomes of, using AI and, using these, precious cells to uncover the secrets. It really has a cost benefit, in the end. These are all very expensive procedures, and I'll talk about mobilized apheresis first.
Okay.
There's no guarantee on how well a donor will mobilize. And when I say mobilize, there's no guarantee on how many stem cells they generate within their bone marrow and that are released into the peripheral blood, which we then capture by apheresis. So, when our clients come to us and they have to go through this process, it's a very expensive process to obtain the starting material. And, the end result could mean that a donor wasn't suitable for mobilized apheresis. So, not only have we wasted that donor's time, but we've also, now passed the expense on to the client to pay for that process. And so, by using AI to determine who would or who would not be a good candidate for mobilized apheresis, there's really a cost savings in the end of all this, because you would, simply guarantee that, the hope is that you would guarantee that a donor would be able to mobilize well and produce a high number of stem cells to then enrich and to be used in the preclinical or clinical setting. When it comes to what Sherry had alluded to about, specifically isolating specific types of cells, one of the projects that we're working on is the isolation of the NK cell and really looking at that, or a natural killer cell. It's also used for therapeutic development. Similarly, as a T cell would be used, as Sherry had alluded to for CAR T. They program these cell types to be killing machines. And when I say killing machines, they kill the cancers that are afflicting, these people. And, one of the things is that we don't understand, from a donor perspective, what makes their cells act in a way or behave in a way or have the characteristics for the manufacturing end of the therapeutic. And this is a very expensive process. It's, hundreds of millions of dollars have been spent on, generating, very specific manufacturing modalities and methods for these different cell types. And so one of the questions from a client perspective that we're trying to help understand is, how well these cells will do in the manufacturing process and how well they'll proliferate or grow and how well the body will receive them once they're put back into a human, either from their own cells or from a donor other than themselves.
Who wants to talk about who you're looking for to become donors at this level?
Donor recruitment is very specialized. So, we often have very specific criteria by which we have to recruit from a pool of donors, if you will. And that could be based off of infectious disease markers. It could be based off of very specific parameters, including HLA type, which generally indicates how well somebody will receive that product from a different donor other than themselves. So it has to be matched. You can recruit based off of ethnicity, demographics, health history, you name it. There's a whole array by which we get asked to recruit very specific donors. So the larger the pool we have of donors, the more we have to choose from to be able to supply these precious cells to these cell and gene therapy companies that are generating products to save lives.
If a person wants to be tested to see if they can donate in this way, what are their first steps?
We have a website, bloodworksbio.org, and you can, there's a fillable form within that website that we would then contact you and pre-screen you. And if you qualify, then you would be inserted into our research donor pool.
Just to wrap things up, I just want to make sure I'm not missing any more salient points.
I just think this is a really exciting way to use data to empower, the way that we work and to empower, us to be, very selective on, who as a donor we choose to go through the process. And it's really just unlocking the power of data and correlating that data back to, our operations. And so I think that there's a lot of power there. And, I'm sure Cherie will back that up. And maybe she's got her own testament to that.
Yeah, I would say on the Ozette side, we're just incredibly excited about this collaboration because what it does is it allows, we have built some very state-of-the-art technology to profile, cells in the body. And then coupled with our machine learning approach, really harnessing, AI machine learning to find signals, to identify people who can respond better, all the things that we've talked about. But it's to us, it's just a great opportunity and a local opportunity for us to partner together to do something that hasn't been done before. And so it's exciting.
It really is. And ultimately leading to saving lives, which is just a beautiful thing. From Ozette Technologies, Cherie Green. She is the Vice President of Translational Science at Ozette. And from Bloodworks Bio, part of Bloodworks Northwest, Executive Vice President Aaron Posey. I appreciate the two of you so much being on the show today and for your collaboration. And everybody, if you want to get in on becoming a donor, go through the process, go to bloodworksbio.org. Is that right, Aaron?
That is correct. Thanks for your time, Lee.
You bet. Thank you.
Bye.